So let's get into the ear. The ear, a lot of people think about of hearing, but the ear has two different aspects. And so if you think of the ear, the nerve that controls the ear, we call it the vestibulocochlear nerve, right? A cochlear implant helps with sound. Vestibule means movement, right? And that's when we're dealing with equilibrium. So we're dealing with hearing and balance. And so there's three parts of the ear that we're going to be looking at here. But understand that hearing and balance, they are very separate once we get into the inner ear, and they act very independently from one another. Okay, so if you look at the ear, we're going to have an external part of the ear, which is here, middle ear, which is this area through here that I'm looking at. And then our inner ear is this snail-like snail-like substance with these little funky shape through here. Okay, so there is our external, middle, and inner ears. Our external ear. When we're dealing with the external ear, the pinna or the auricle is the outer part, which helps bring sound into the external acoustic meatus, which is going to help bring it to the tympanic membrane, also known as the eardrum. In this tube, not only does it bring sound through here, but it also creates cerumen, like a wax, right? The ceruminous glands are in there. And then we come to this middle ear. The middle ear is separated by this tympanic membrane. So the tympanic membrane separates external from internal ear. The external to the middle ear. The middle ear is an area that's filled with air. This is not fluid in here. It's a small air-filled sac, and it is laterally connected to this eardrum, also known as the tympanic membrane, and more medially to the inner ear, it's connected by what we call the oval window. And there's three separate bones, the smallest bones in our body. We have the malleus, the incus, and the stapes. Those are the three different bones, and that's going to bring it to the inner ear. In our inner ear, we have what we call the bony labyrinth that we're going to be looking at. Okay, now another thing you're going to notice on this middle ear is this auditory, also known as the pharyngotympanic tube, also known as eustachian tube. What it does is this is going to go into our throat, and this will help bring air into this area. So as we go up and down elevation, we create equal pressure on the inner part of this middle ear and the outer part of the external ear. There's an equal pressure on both sides of this eardrum because we want the same pressure in this area to create optimal vibration of this eardrum. And that's going to create sound, and that's done through this auditory tube. All right, so let's look at these structures a little bit more. So we have the outer ear. Our function of the tympanic membrane is to carry sound from the external source and bring it into the middle ear. That's the point of this, its function. And the function of that is to make the malleus, incus, and stapes move and vibrate. So it's to transfer sound energy from an external thing, transfer sound energy onto the small bones of the middle ear. Okay, that middle ear that is flanked laterally by the eardrum, medially by the oval and round windows, which we'll look at in a moment, and then more that inferior aspect, we have the auditory eustachian tube. The function of this is to create equal pressure from the external ear and the middle ear so that that eardrum, the tympanic membrane, can move optimally to transfer that energy into those small bones. And then that brings us to those malleus, incus, and stapes. Those are three separate bones. And what they do is they're taking that outer sound energy from the external ear that gets transferred to them and amplifies the vibration So it does so, it amplifies it and sets into motion sound waves into the inner ear. These sound waves. And so this is going to be, it causes these to amplify the sound. And on the other side of this bone, there's a thin membrane. And as this moves in and out, it's going to put sound waves into this area through here, the cochlea. Now, those sound waves, the way they get transferred is there's actually fluid in here that we're going to look at in a moment, and it sets little motion into the fluid of the inner ear. Now, let's look at this inner ear. So I said this sets into motion through the cochlea, but we have the cochlea, which is the snail version. This is where hearing is going to happen. And then we also have this area, which is our vestibule and semicircle canals. This is the vestibular portion dealing with equilibrium and balance, sound. Now you'll see here, there's two branches of this nerve. It almost looks like two separate nerves. We have the cochlear nerve and the vestibular nerve, but we didn't separate those nerves when they came out of the cranial nerve, did we? We call it the vestibular cochlear nerve. So sometimes you'll see it called the cochlear nerve. It's more accurately the cochlear division of the vestibular cochlear nerve. This is the vestibular portion of the vestibular cochlear nerve. That's a little bit more accurate way to say that. But same concept, right? These are the nerves that are going to take equilibrium and balance and send information to the brain. This is going to take sound information and send that to the brain. So let's look at that inner ear. Now, the outer portions are what we call bony labyrinth, and inside that bony labyrinth is a membranous labyrinth. So if I looked at that and saw through it, here it's all bony. This is all bony structure, bony labyrinth. But inside of that is floating a membranous labyrinth. And if you look, what we're going to find is this membranous labyrinth, yes, it has a membrane that is filled with the fluid, but there's also fluid between this membranous labyrinth and the outer portion of the bony labyrinth. So there's some fluid in this region and a different fluid in this region. It is this membranous labyrinth that we're going to stimulate sound and balance information to our brains. Okay, so let's look at this. Here, let's look at the vestibular portion first. Here is the vestibule. So let's go back to the bony labyrinth. The bony labyrinth contains worming their ways all the way through the temporal bone, and it contains the cochlea, the vestibule, and the semicircular canals. And it's filled with what we call perilymph. So here is our cochlea, our vestibule, and the semicircular canals. Three different things. Now, when we get down into the membranous labyrinth, there's a few more. The cochlear duct is sitting in the cochlea. And remember, inside the bony labyrinth is perilymph. Inside the membranous labyrinth is endolymph. So sitting in the cochlea is this cochlear duct. Sitting in the semicircular canals is semicircular ducts. Now inside the vestibule, there's two sacs. One's called a utricle and one's called a saccule. The utricle and the saccule are going to be dealing with more linear movements of balance and equilibrium. This is more dynamic twist turns, which we'll look at in a second. So let's look at that now. Here's the cochlea, and inside the cochlea duct, right? Here is the semicircular canals in the bony part, and sitting in there are the semicircular ducts. And then here's the vestibule, and you can notice there's one sac, the utricle, and the second sac, the saccule. Okay, two sacs sitting in there. Now, suspended in that perilymph is this vestibule are the saccule and the utricles. The saccule extends into the cochlea, the utricles extend a little bit more into the semicircular canals. And what they're going to do is they're going to measure different responses. And I think there's, yes, the utricle is going to measure what we call horizontal movement. and saccule vertical movement. So horizontal side to side, vertical up and down. And so that's what those two things do. So how do they recognize and share that movement? That's where we're going a little bit, back up a couple slides. All right. So they are going to be, these sacs house receptors, and those cluster of receptors are called the maculae. The maculae respond to these linear movements, whether it's horizontal or vertical. And how does it work? Well, all of these movements you're going to see here, whether it's hearing, whether it's equilibrium, balance, they're all dealing with little hair cells, the hair cells, which some of you may have heard that these little hairs in our ears, what makes us hear. It's the same thing with balance and equilibrium, right? All right, so in these areas, we have the macula, and in the semicircular canals, those areas that house the receptors are called crista ampullaris. They're really similar in how they work. You'll see it's all about some supporting cells, some hair cells, and then the hair cells move to stimulate the registration of vertical, linear, and dynamic movements. You'll see how those work, okay? So what does it look like? Well, let's look at the maculae first. The maculae are this group of sensory receptors, and they're going to contain what we call supporting cells and hair cells. Each of these hair cells has an extension of actual hairs called stereocilia, and they're going to be embedded into a jello-like membrane called the otolithic membrane. So here's what it looks like. Here are here's that whole area, the maculae. They sit at the bases of the utricle and saccule here. And we have a bunch of supporting cells. And then here are those hair cells. The hair cells have hairs that come out of them. Those hairs are called stereocilia. And they get embedded in this membrane here, which would be covering all of it. We just cut a little portion so you can see it. And it's really like jello. It moves and jiggles around. It's not completely solid. It's in a solid state, but it moves. It's not rigid, I should say. It's more of a dynamic jello-like substance. And then on top of that jello-like substance are a bunch of little otoliths, which are just like they sound. They're like little rocks. They're little weighted substances, otoliths, little rocks that sit in that movement. And what will happen here is as we move this membrane, if we move it to the left, this bottom membrane is going to move faster because of this weighted hair follicles or weighted otoliths sitting in this membrane. And then all of a sudden, that movement being slowed down a little bit will stimulate these hairs to move. So what does it look like? So here's my basement membrane through here. Here's my the jello-like substance that those hair follicles move. I move, and then all of a sudden the jello follows. I move, and you can see it moves first, and then it measures that movement. The hair has now moved. It stimulates those hair follicles and then follows. So it's moving like this. And even if we laid on our side, they would slide down a little bit because of the weight through here. So we can move, and that movement of the base being different from the otoliths on top of that jello-like membrane, they're going to be moving at slightly different times. So that's what's going to stimulate those hair follicles to tell what movement is. Okay, so here we go. Let's look at that again. We have this move, and then this will be delayed a little bit and cause these hair follicles to be stimulated. That sends a signal to that part of the vestibular cochlear nerve, the vestibule portion of it. So that's linear movement. And when we're dealing with that linear movement, the utricles are going to respond to the horizontal, saccule, vertical, but it's the same concept of how we move, and it stimulates us. Okay? Now, the crista ampullaris, it's a little bit different, but it's still the same concept, right? We have hair follicles that are going to be moved, and when they move, they're going to stimulate the nerve to tell our brain that we have moved. But it does so more in this dynamic equilibrium. It's not just these linear movements, the bends, twists, rotations, those kind of things. And here's what this looks like. So here are these semicircular ducts sitting in the canals, right? And at the base of those areas, we have this cupula. The cupula sits on top of a membrane, and this membrane has hair cells and supporting cells, and those hair cells have stereocilia that come up into this cupula. And this is all fluid. So now, if I move in a linear way really quick, the fluid doesn't move as fast, and so all of a sudden we'll see the hair move. So say I moved quickly this direction, well, this kind of stays still, or the fluid stays still, and so the hair follicles move the opposite direction on that. It stimulates that. Now, I talked about rotation. Well, what happens if we keep rotating in a circle? Well, the more you move, eventually this fluid will start moving with you. And that's what happens when we spin. So all of a sudden, this person starts spinning to their left. They start spinning, and then all of a sudden, that fluid stays still at first. So when we move, that hair follicle moves. But as they keep spinning, all of a sudden that flow will start going with us. We're not noticing it as much until we stop. But now that fluid is continuing to move and will cause us to still feel like we're moving, because all of a sudden the membrane has stopped, but the fluid is moving. Whether it's the membrane moving and the fluid stopped, or the fluid moving and the membrane stopped, that is moving these hair follicles, which is telling us that we're moving in a dynamic equilibrium way. Now, depending on whether it is the maculae being stimulated through linear movements, or the crista ampullaris being stimulated, what happens is those nerves from the hair follicles, that's part of the vestibular portion of that vestibulocochlear nerve, well, it's called the vestibular nerve right here, that is going to send a signal. So here's what looks. We change positions, otoliths within the automatic membrane, or the cupula shifts position. That's going to cause those hair cells to be stimulated because the stereocilia have bent. That causes a depolarization and activation of the vestibular branch of the vestibulocochlear nerve, cranial nerve 8. That then goes to our brain, specifically goes to our cerebellum first, because that's dealing with balance. And it also affects our eyes. Affects our eyes, as well as cranial nerve 11. Cranial nerve 11 through here is dealing with movement of helping with trunk limb movement as well, spinal motor control, we call it. So let's look at this. We haven't talked about the visual receptors yet. We're just dealing with the vestibular portion. The vestibular can stimulate the cerebellum, which then can stimulate the brainstem, the vestibular portions of that, right? And that is going to affect our eye movement. That's why when we get dizzy, our eyes try to move up with that, as well as the vestibular cochlear nerve coming in here, as well as cranial nerve 11. Now, we can also see it goes in and goes directly to the brainstem. So some information goes to the cerebellum, some goes to the vestibular portion of the brainstem. That's where we're seeing vestibular branch and cerebellum. Yep. And that's our equilibrium shifts and change. Now let's look at hearing, sound. This is also done through hair follicles, or hair cells. And these hair cells are going to measure movement of fluid, just like it did in the vestibular balance equilibrium portion, in a slightly different way. But it's still dealing with hair follicles being stimulated and causing depolarization, but this time it'll go to the cochlear division of the vestibular cochlear. Here's what this snail-like cochlea looks like if we cut it in half. Here are those membranous labyrinths sitting in the bony labyrinth. And you're going to see 3 different chambers here. Those 3 different chambers are, let's see, where do we have? The 3 different chambers are scala vestibuli, scala tympani, and the cochlear duct. Now, the scala vestibuli and the scala tympani, they actually are connected. How do I show this? We do not see connection here, but if we took, imagine this cochlea and I started to unwind, and unrolled it, it would be this long process. So you can actually see I'm starting to unroll it. You can see this scala vestibuli and the scala tympani, they're on opposite sides of each other, but they will eventually connect at this very top portion through here. This is that top portion, okay? So that's why we're looking at this. They actually connect at one point. Both of these are filled, they're both filled with perilymph. So this is all perilymph. And if you remember from the vestibular portion, the membranous portion had endolymph. Well, here we have this inner portion. This inner portion we call the cochlear duct, and the cochlear duct is filled with endolymph. So, scala vestibuli, it begins at the oval window, contains perilymph. Scala tympanis is continuous with the scala vestibuli way at that top portion and ends at the round window, and it also contains perilymph. The cochlear duct, this is where the sound receptors are. And specifically, that group of where the sound receptors are is called the organ of Corti. And the organ of Corti, you're going to see some hair follicles. Now, this whole duct contains endolymph. So now let's look at this. We're going to be looking, here's our cochlear duct, and then through here, that's going to be our organ of Corti. So let's take a little bit closer view at that. Scala vestibuli, scala tympani, and sitting in between here is the cochlear duct. There is this basement membrane that sits between the cochlear duct and the scala tympani. And if you look at that even closer, here is this picture. Here's that scala tympani down here. This whole region through here is our cochlear duct, and here is the organ of Corti. Supporting cells, hair cells, stereocilia coming in to the tectorial membrane. And if we get movement in these fluids, it will cause this tectorial membrane to move, which actually moves these stereocilia of the hair cells, and that gets stimulated and travels down this cochlear division of the vestibulocochlear nerve, creating sound. So, how does this area, this endolymph of the cochlear duct, get movement? How does this tympanic membrane start getting movement in here? Well, here's what it's going to look like. Sound vibration is going to beat against our eardrum. We'll come here. Beats against the eardrum. This malleus, incus, and stapes intensify that sound, and this is the oval window. This oval window pushes vibration in the water, in this fluid, the perilymph, and it moves along through here. Now, if it is a what we call a high frequency, it's going to transfer closer to the closer to that middle ear, closer to what we call the oval and round windows, versus if it is a lower frequency, it'll move a little bit higher up through here. And what will happen is this is really stiff through here. So this membrane is very stiff, and the fibers, the hair follicles are shorter. So all of a sudden we're having it pretty stiff through here, but we're going to get to a point where all of a sudden it's not as stiff and we're able to allow sound to move through here. Sound vibration pushes into that center cochlear duct. And once that pushes in through here, that gets this tectorial membrane to move. Higher, it's more like a harp. If you look, that's why it shows it like this, where our bass are shorter fibers, it's stiffer, they're more of a higher frequency versus lower frequencies, though they're not as stiff, longer fibers. And so this is stiff, stiff, stiff. All of a sudden, when it sounds low enough, it'll be able to get through here. If it's too high, it won't be able to. And that's how we dictate sound. And depending on what part of this gets stimulated, that's the information that gets to our brain to tell us if it's high and low frequencies, and we distinguish sound that way. Now, if the frequency is really low and it's so low that we can't hear it, what can happen is this vibration can go all the way around and it will never stimulate the cochlear duct and our organ of Corti. Also, if it's too high, we can't hear it because it just doesn't even get very far through here. Yeah, that's where we're dealing like with dog whistles, right? They're a frequency that we can't hear. That is going to send that information to the cochlear division of the vestibulocochlear nerve. That information goes to the medulla, which then goes to the inferior culiculi, which then gets the thalamus to be passed on to the auditory cortex, which is in our temporal lobe. Yeah, here's a good picture of that. Stimulates through here to the medulla, midbrain, inferior culiculi, goes to that thalamus region, and then to the temporal lobe for processing of what we're actually hearing. And that is sound. Yeah, I think that's all I have for sound-wise on that. Okay, so then that brings us to the last couple sensations, and these are gustation and olfactory. Olfaction, gustation, taste, olfaction, smell, and both of those are dealing with chemical receptors. And they are a chemical substance, which in the taste, when we're dealing with gustation, that chemical substance is going to get dissolved into saliva and picked up by our taste buds. Smell, that substance gets dissolved into the mucous membrane of our nose and gets picked up by the olfactory nerves in our nose. Sense of smell. The organ for the sense of smell, we call it the olfactory epithelium. So you just have numerous different cells that are sitting on in our sinuses through here, and here's our cribriform plate. Remember all those little holes that sit in the ethmoid bone? And you have some nerves that are going to be coming in through here and being picked up in the mucous membrane. Look what kind of nerves these are. Again, bipolar nerves. There's two arms to this bipolar cell. So we have some bipolar cells picking up the chemical sensation, sending it to the olfactory bulb, and that olfactory bulb then takes that information to the brain and to the olfactory cortex. The olfactory cortex is sitting in our temporal lobe as well. But we're also taking some of that information and bringing it to the frontal lobe, and that's where we can recognize what we're smelling. And it brings us into the amygdala limbic hypothalamus situation. This is emotion, and that's why smells tie into emotions quite a bit. Smelling home-baked cookies reminds us of our grandmother. If we were ever in a fire and people were injured, like that smoke would raise alarm systems in our fear center based on past experiences. So smell will also affect memory, emotions, as well as you know that it stimulates our stomach secretions, all kinds of things. So we're going to be having signals go to olfactory in our temporal lobe, frontal lobe, and then the limbic amygdala hypothalamus areas. And smell, we're dealing with, again, chemical receptors, chemoreceptors. There's about 10,000 or so taste buds found on our tongue. and those taste buds are found in our papillae. Now, papillae are more of the bumps, and not all of them have taste buds. Most of them do, but the filiform, which is the majority of all the bumps on our tongue, the filiform, they do not have taste buds. However, filled in between all of these filiform papillae are the fungiform. And here are those, the fungiform, and those are a large majority of our taste buds. We have these little slits on the sides. Those contain taste buds as well. We call it the folate papillae. And then these big giant circles on the very back of our tongue, we call them the circumvallate, sometimes the vallate papillae. Usually around 8 to 12 of those on people's tongues, and those also contain taste buds. Taste itself has 5 main tastes that we feel experience: sweet, sour, bitter, salty, and umami, which means savory. All of this information then gets traveled to from various different nerves. We're dealing cranial nerve 7, 9, and 10, dealing with different parts of our tongue. Remember, it's not just one nerve that goes to all of our tongue. We have different nerves that go to different parts of our tongue. So these are the 3 nerves that are going to be dealing with that taste, and they'll send it to the medulla, and then from there it'll go to the thalamus to process, and then eventually arrive at that gustatory cortex. The gustatory cortex is in the insula, but you can see here it also deals with that limbic hypothalamus, and taste can also bring in certain emotions and memories. That's what this shows us